Hi, my name is Brett Benkowski, and over the last couple weeks we've done a series of assays investigating methyl jasmonate and its effect on human leukemia 60, better known as blood cancer in some circles. And so we've done assays on viability and cell morphology to evaluate whether or not methyl jasmonate is a potential cure for leukemia in humans. And this molecule on screen is methyl jasmonate. It's different from jasmonic acid because of this CH3 group, that is the methyl in methyl jasmonate. So first we're going to give some brief background on HL60 and why we use it. So human leukemia 60 was first established in 1977, and it was taken from a patient with acute myeloid leukemia, so a form of the blood cancer. The cells resemble promyelocytes, which are here on the blood cell differentiation tree, and they're cancerous because they're stuck in a state of permanent division. They never differentiate into what they're supposed to be. So over the years we've found that different reagents can make HL60 become the next stage in the cell cycle, and that breaks it out of the cancer cycle. So it can become granulocytes or monocytes, and those will eventually go on to develop macrophage activity. And we also know that the cells stop being cancerous at this point because the HL60 cell contains an oncogene, which is the gene that codes the cancer activity, but that gene disappears after cell differentiation. In addition to causing the cell to differentiate, the other way to break it out of its cancerous behavior would be to make it go through apoptosis, or programmed cell death. So in apoptosis, the cell shrinks due to the pro-apoptotic trigger, which causes the apoptosis, and it goes from its healthy state to starting to expel liquid, condense, the DNA and nucleus condense here, and then it fragments, and then the cell itself breaks down into apoptotic bodies. And that process is also called blebbing, and the important part there is that the cells are never broken open, so the cytotoxic material stays within the cell. From there, these apoptotic bodies are fed into the macrophage, where they're broken down. In order for a treatment to be considered useful, it's important that the cells undergo either differentiation or apoptosis. If the cell undergoes necrosis, which will be characterized by the cell membrane dissolving and the cytotoxic components leaking out, then the treatment should probably be avoided because it exposes the cellular environment to unnecessary cytotoxic elements. The first experiment we did was a standard viability assay. So using controls involving the media that we culture the cells in, the media and the cells, media cells in 70% ethanol, and the media cells in camptothecin, which is an antibiotic that's been known to induce apoptotic activity, we were able to see how viable the cells remained. Cell viability was measured using absorbance and the WST8 solvent, which is a solvent that turns orangish red when it's reduced, and only active viable cells can reduce WST8. So higher absorbance values indicate a stronger red color, indicating more viable cells in that well. The viability assay indicates that methyl jasmonate has the capability of reducing cell viability both with and without extraction in 70% ethanol. Methyl jasmonate comes as a liquid, but as part of the standard procedure for the experiment, the substrate should be extracted in 70% ethanol, so a mixture of 70% ethanol and methyl jasmonate was put together in order to maintain continuity, but pure methyl jasmonate was also tested. It was found that pure methyl jasmonate reduced the cell viability more than methyl jasmonate with ethanol in it, and that's probably because the methyl jasmonate with ethanol in it was diluted as opposed to the pure methyl jasmonate, where there was more methyl jasmonate in the sample that was used to culture the cells. Another assay we used was the fluorine fluorescence morphology assay. So cells were treated with HL60, which attaches to DNA and makes it glow green when exposed to fluorescence. So using the fluorine laser microscope, we were able to see that healthy cells were observed in the presence of ethanol. So cells like this, where the green is not concentrated but is spread out, and this one are good examples of healthy cells. Whereas over here in the camptothecin cells, cells like this one and this one and this one, where the green is condensed and concentrated, indicate that the DNA has started to condense and the cell is undergoing apoptosis. For an experimental sample, we used the fluorine microscopic morphology assay to test pure methyl jasmonate at 48 hours. So we did not see very much condensed DNA here. Cells like this and this appear healthy. This is a brighter green, which could indicate that maybe there's some condensation going on, but overall these cells appear to be pretty healthy. So cells treated with methyl jasmonate after 48 hours do appear healthy. This means that methyl jasmonate could have a very temporary effect on human leukemia, decreasing the viability over the short term, but over the long term disappearing in effect. The morphology assay was repeated for samples at 60 hours, and while in that time some of the fluorescence wears off from cyto16, we were able to isolate one cell for both pure and ethanol extracted methyl jasmonate. So in the methyl jasmonate extracted in ethanol, the cell appears to be more condensed than the cell in pure methyl jasmonate. This assay ended up being pretty inconclusive because we were only able to visualize one cell for ethanol extracted in pure methyl jasmonate each. However, for both of them, the cell appears to be pretty condensed. The one in pure methyl jasmonate appears less condensed. So most probably, we just caught these cells at different points during the apoptotic process because we know that methyl jasmonate can induce apoptosis. So what appears to have happened is that we found cells that were undergoing apoptosis related to the methyl jasmonate. Whether that's due to luck or because the methyl jasmonate, it does have a lasting effect, is undetermined because we only have one cell to examine. The last assay we did was called a COMET assay, sometimes known as a single-cell electrophoresis assay, where we electrophoresed spun down samples of the HL60 treated with pure methyl jasmonate. So here is the one comet that we were able to visualize with the dense DNA here and then the tail approximating this shape. So what this tells us is that the methyl jasmonate was able to cause damage to the cellular DNA, which means that at some level, methyl jasmonate does affect cancer. So to conclude, methyl jasmonate was able to affect human leukemia 60. It significantly reduced the viability of HL60 cells after 24 hours, but did not conclusively show morphological evidence of apoptosis at 48 or 60 hours. It did show common assay evidence of damaging DNA, so the logical future step to take in this research would be to determine how long methyl jasmonate affects cells for. If it ever was made into a drug, this active period would influence how often the drug would need to be administered. Also, it would be good to pursue a way to make methyl jasmonate miscible in water because methyl jasmonate is strongly nonpolar and water is polar. So it does create oil droplets in the water solution, which would cause difficulty in administering it as a drug and also may contribute to the reason that methyl jasmonate is not effective at long periods of time. If anyone wants to follow up on any of the research, or most of these are just actually image citations, but if anybody wants to check up on any of them, here are the citations listed in order of appearance and reference. I also just want to specially thank Professor Maxon for taking pictures and sending them to me from the fluorescence microscope. Other than that, thank you so much for your time and have a great afternoon.